Oh, quite the opposite actually. Thank you. Thank you. Thanks a lot. Yes. No, but our exposure was we use the plus to be able to create. It's an out of the box. Some places. Yeah. No, no, I grew up in, I was born and raised in India, so I say this I mean, this winter was the first time I saw this. So, I mean, I've been to places where there's been some weather, I have quite a lot of research until then when my parents were visiting, it's obviously very popular tourist destination. Yes, yes, they were here last year. It was, yeah, it was nice, nice vacation. Yes, yes. Yeah, you should amazing, because it gives us everywhere is very similar. The winters, like this winter, it's very diverse. I would definitely be recommend this. Yeah. Yeah. Yeah, yeah, absolutely. I mean, it's
I know.
we never felt, I mean, we were living like this, never felt that also everything is over.
We know better.
I mean, I don't think they would shift everything. They have such a strong support system. Yeah. No, no, I'm the only child. But family. They eventually down the road, I guess. Again, because I'm we did only music for a bit, but it's the happy first, I guess. Yeah, it's a lot. It doesn't make sense to go for a few days. Yeah, yeah, she was. No, no, she moved. She's been living since she got her master's here. She works in her yes. So she's more than a creator as this called. She works at the tax. That's very true. And it's such a new field, like I don't. No, she was getting her degree. Yeah. Although now they are using a light job. Oh, I see. Yeah, I mean, I think it's one of those things that I think everybody.
Good afternoon, everyone, and welcome to PIKNIC. How many of you have been to a picnic before? Great. That's that's super. My name is Sarah McIntosh, and I have been around MD Anderson for quite a while. Somehow I feel like I'm so short that I can't see you, but I know you're there. So I am a member of My Cancer Connection and have served in various capacities in that with that structure that's so helpful and wonderful to patients and caregivers here. And I'm also a 22-year survivor of breast cancer. So I'm a great fan of MD Anderson. I want to welcome also our participants who are viewing this remotely at the centers in the Bay Area, Katy, Sugar Land, and the Woodlands, and those watching remotely as well. So as you know, PIKNIC is one of the many programs of the My Cancer Connection part of volunteer services and merchandising at Anderson. And it's brought to you in an effort to enrich the survivorship programming and generously funded by the Volunteer Endowment for Patient Support and the gift shop proceeds. My Cancer Connection's mission is to share hope, support, and understanding to all those who are challenged by cancer from those who have been there. More information about My Cancer Connection programs can be found on the program cards at your seats. Now, before we begin the program today, I'd like to bring to your attention that the session is being recorded by UT Television and will later be available on the My Cancer Connection website. If there are those of you who prefer not to be in the camera range, you can just move down to that end of the room, and that will keep you safe from the lens. But it is my pleasure now to introduce today's MD Anderson expert. Dr. Gopalakrishnan is a translational scientist who is committed to advancing the treatment of cancer through iterative studies in mouse and man. He is a trained dentist who obtained his degree from Bapuji Dental College and Hospital in India in 2010. He also has a PhD in cancer epidemiology, during which time he joined the laboratory of Dr. Jennifer Wargo at the University of Texas MD Anderson Cancer Center to lead studies in characterizing the human microbiome. Currently, he's working as a postdoctoral fellow in her lab. Dr. Gopalakrishnan's work focuses on better understanding differential responses to cancer therapy through analysis of the oral and gut microbiomes with parallel studies in matched tumor and blood samples, as well as in murine models. In a cohort of anti-PD-1 treated melanoma patients, he has helped identify differential signatures in the diversity and composition of the gut microbiome in patients who respond compared to those who did not. Dr. Gopalakrishnan has been incredibly productive and has presented his work at several international meetings. His goal is to become a leader in translational research. Please join me in welcoming Dr. Gopalakrishnan to today's session.
Good afternoon, everyone, and thank you very much for that generous introduction. It is truly an honor to be here today and to share with you our insights on what we think is the role of the healthy gut on influencing our immune systems. Here are my disclosures. So the study that we did was in a cohort of melanoma patients. So I thought I would begin this talk by briefly introducing melanoma. So melanoma is a type of skin cancer which affects approximately 3.5 million patients in a year. It's a therapy-resistant tumor of melanocytes. These are cells that give our skin its color and are pigmented in nature. So, and their most common etiology is exposure to ultraviolet radiation from the sun. There are several different types of melanoma, but the most common presentation is the cutaneous type, which occurs on our skin surfaces. We have certainly made major advances in the treatment of melanoma and other cancers through the advent of targeted therapy and immune checkpoint blockade, with several new agents being approved for use by the FDA over the last 10 or so years. Certainly, therapeutic targeting of an oncogenic mutation leads to regression of melanoma tumors. Here I show you a PET scan of a patient with melanoma tumors all over their bodies. But after treatment with a BRAF inhibitor, these tumors have almost completely regressed. Another major breakthrough in the treatment of cancers has been the advent of immune checkpoint blockade therapies. These basically act by unleashing the brakes on our immune system and allowing our body's own immune cells to attack cancers. They have represented a significant landmark in our progress towards eradicating cancer. And as we see here, several patients achieve long-term disease control. However, in the same plot, one can also see that these responses are heterogeneous and not everybody responds. Therefore, the next obvious question that we have is, how can we better understand these responses to therapy and how best can we optimize these treatment regimen? We certainly know that several factors, both at the site of the tumor as well as in the systemic circulation, affect cancers. But what we are also beginning to learn is that environmental factors contribute significantly towards this phenomenon. And the microbiome, which is going to be the focus of my talk, is a significant component of these environmental factors. So what is the human microbiome? It can be thought of as the sum of all microorganisms that inhabit our human body. They actually outnumber our own cells by a 10-to-1 ratio and constitute up to 3% of our body mass. Here is a brief bacterial primer, and I apologize for this segue into 9th grade biology, but I do transition through several of these bacteria during my talk. So standard bacterial taxonomy is arranged from phylum, which is a larger group of bacteria, to a more specific group called genus or species. So for example, within the Firmicutes phylum is the Clostridia class, the Clostridialis order, and the Faecalibacterium genus. The fact that we can study the microbiome today is largely attributable to the efforts of two large consortia, the HMP, or the Human Microbiome Project in the United States, and the MetaHit consortium in Europe, that provided us tools to comprehensively and cost-effectively characterize these bacteria in normal adults, and thereby providing a critical baseline on which future studies could feed off. In these studies, up to 18 different sites were characterized in the human body. These range from 9 different sites within the oral cavity to the gut as well as skin. But the largest and most active collection of microbes is in the gut. Hence, that is what we decided to focus on too. The bacteria in the gut, they continue to evolve over time and are shaped by many factors that take into effect even at birth. So at birth, actually, the diversity of and the number of bacteria are very unstable, and they gain in number over time through the first year of life, after which these numbers reach a plateau. The actual bacteria itself are influenced by several factors such as diet, genetics, hygiene, and lifestyle. In fact, even route of delivery of a newborn influences the type of microbiome that it gets. These bacteria in our body are not passive at all. They in fact very actively interact with our epithelial and immune system and perform several important functions. Primarily help in digestion because they increase the metabolic capacity of the host and digest otherwise unused food components. They help in completion of another aspect of our digestive cycle, and also prevent the colonization of harmful pathogenic bacteria. So how can we characterize the microbiome? I will try not to get too much into the technicalities of this, but basically there are two commonly used methods. The more frequently used is the 16S approach. The 16S is a ubiquitously found genome bacterial marker. It is present in all bacteria. So one can look for this gene, amplify it, and then identify the bacteria that are present in the communities. There are also several reference databases against which we can map our findings to confirm the bacterial taxonomy. It also gives a sense of how diverse a population is. So more diverse a population, greater the bacteria there are. A more sensitive approach is the whole genome shotgun sequencing approach. It's more costly, but it also affords greater resolution of bacteria. So we are more confident in the type of species that we identify through this approach. In addition, it also gives a sense of the function that these bacteria perform. So in addition to knowing which type of bacteria are there, we also get a sense of what they do. So again, here are some commonly used metrics in microbiome studies. So diversity you hear me constantly say it, is of two types. It's alpha and beta. So alpha is diversity within a sample, and larger the number, more the bacteria in that sample. Beta diversity helps compare two samples. So these can now be visualized in space to see how closely related they are. What is more interesting though is to identify the bacteria in a sample. So here, for example, is one patient sample, and every color within that sample in one column represents a different bacteria. So we get a sense of not only how many there are, but how many of each there are as well. So can these bacteria actually help shape our immune responses? Our immune system originally evolved to fight invading pathogens, but we have learned over time and learned so quite strikingly that these can also fight cancer. Here is an illustration just to show you the complex interplay that takes place between bacteria and the immune system and how closely the two systems are linked. So under normal conditions, homeostasis is maintained through regulation of tolerance between the bacteria and the immune response that is mounted against it. Under these situations, everything is fine. But disruption of this balance leads to a condition called dysbiosis, which leads to several adverse effects. So what is the role of microbiome in disease? It is very apt now to invoke Hippocrates, who several centuries ago mentioned that all disease do begin in the gut. And it is proving now true when dysbiosis has been found to be associated with several different adverse conditions ranging from diabetes to even depression. In fact, there's also growing appreciation of the role of the microbiome in cancer development. And as you can see, there's been an explosion of evidence in this regard, with several investigators pursuing this field actively. It has also become very clear that the microbiome is closely linked to influencing differential responses to cancer therapy. Much of this work, though, was done in the setting of stem cell transplants initially, where investigators found that high diversity of the gut microbiome at the time of the transplant was associated with improved survival. So let me go over this again. So the high diversity, in high diversity patients, you can see that there are many bacteria here. When you compare them to intermediate diversity, where you can see fewer, and low diversity, where you can hardly see any bacteria. Each color again here represents a different bacteria. This is a commonly plot survival curve. It shows the probability of survival on the y-axis. So the higher the line is on this axis implies better survival. So patients that began with high diversity, you see, have a much higher probability of survival in this group. But certainly, in addition to diversity, individual bacteria also matter. And there were several seminal papers that were published a few years ago that implicated specific bacteria within our gut that could impact therapeutic response. So armed with this background, we next asked, what is the role of the microbiome in metastatic melanoma patients who are undergoing treatment with immune checkpoint blockade? We initiated this study about 3 years ago and now have more than 300 patients enrolled in our cohort. A oral and gut microbiome sample was collected from these patients before they started therapy. And if possible, we also collected a repeat sample over time in order to assess if there are any changes in the microbiome because of the therapy. In order to perform correlative studies, matched tumor and blood samples were also obtained from their patients at pre-treatment time points. As you can expect from an institution like MD Anderson, a wide variety of treatment types were represented, but the largest and most homogeneous subset were those who were treated by this anti-PD-1 therapy. Hence, we decided to focus our analysis on that cohort. All microbiome samples were subject to 16S sequencing, which I just introduced, and a subset of them to whole genome shotgun sequencing. Tumor and blood samples, we did deep immune profiling to understand what type of immune cells there are in these patients, using a whole host of assays that are listed here. So our hypothesis going into these studies was that differential bacterial signatures exist in responders versus non-responders to immune checkpoint blockade, that favorable signatures are associated with an enhanced systemic and anti-tumor immune response, and that insights gained from these studies could lead to therapeutic optimization through modulation of the microbiome. So, as I mentioned, we collected samples from 2 different sites. One of the sites we chose was the oral microbiome. In order to do this, we swabbed the inner cheeks of these patients. This was very easy to do, so we did this while the patient reported to clinic, and we had fairly high compliance rates on this. Almost every patient enrolled, we have a buccal sample from them. The fecal samples, on the other hand, were collected on an outpatient basis. So the patients were given the kit when they visited the clinic, and then they took it back home, collected the sample in the privacy of their homes, and then mailed it back to us. The kit had a buffer that allowed for stabilization of these bacteria for up to 2 months at room temperature. So our first goal was to just look at what we have in these samples as to what the distribution of bacteria are. As you can see, both in the oral and gut microbiomes, there's a large diversity in terms of the bacteria that are present in this population. However, you can also appreciate that the Lactobacillus group and the Bacteroidealis group predominated in both the oral and the gut microbiomes. Next, we sought to answer if there is any association between the oral and the gut microbiomes. But as you see here, these 2 groups cluster almost completely separately, thereby suggesting that these are not really representative of each other, as one would expect, and that one cannot use one site as a surrogate for another. So their activities are quite different. So again, the first question we asked was, is there an association between diversity of the microbiome and response to anti-PD-1 immunotherapy in this cohort? To do this, all patients that were enrolled in our study were classified into either being a responder or non-responder based on routine X-rays that are collected from these patients and are assessed periodically. So interestingly, patients who did respond had a much higher diversity of their gut microbiome at baseline compared to those who did not respond. This was in the gut microbiome again, and we did not see any differences in the oral microbiome. So next, we classified our patients based on their diversity scores as having either high, intermediate, or low diversity, like our collaborators had done from the stem cell group. And we found that those that had high diversity, again, you can see from this curve that they did much better while on therapy compared to those that had intermediate or low diversity. So therefore, high diversity of the gut microbiome was influential of response, and this diversity was assessed before treatment. Another metric, like I mentioned to you, was beta diversity. This gives a sense of how similar two samples are. So again, from this plot, it's quite evident that responders and non-responders differ quite significantly in their compositional structure. So we know that there are some differences in terms of their components on comparing those who respond and those who don't. So the next question we asked was, what are these compositional differences that exist between, in terms of bacteria, that exist between patients who respond and those who don't? So here is a complex plot, and I probably won't go into too much detail, but this is basically made using a tool from a group up at Harvard. It helps identify what bacteria differ between groups and also assigns a particular effect to them. So in this plot, you see here that longer the length of the bar, larger the effect that is associated with that bacteria. Also in this complex graph, which is called a bacterial cladogram, is arranged from a higher level, such as phylum on the outside, and as one moves further down, it is more specific. So right at the center, you would have species. So blue represents the bacteria that are enriched in responders, and red represents bacteria that were enriched in non-responders. So interestingly, we saw that the Faecalibacterium genus within the Ruminococcaceae family was significantly enriched in responders, whereas the Bacteroidalis order was significantly enriched in non-responders. So having seen this, we obviously then we looked at each patient and asked how much Faecalibacterium they have. So we also classified patients therefore as having high Faecalibacterium in their gut or low Faecalibacterium, and saw that patients that had a high abundance of this bacteria did much better on therapy than those that had a low abundance of bacteria. This is a subsequent assay that we did to confirm these taxonomical differences. You see here that again we saw Faecalibacterium show up as being differentially abundant in responders compared to non-responders. In fact, it is significantly present here, whereas hardly present here at all. It also gave us a sense that these two groups, the blue being response and red being non-response, they grouped quite differently, and suggesting that the actual, not only are there differences in the type of bacteria, but there are also differences in the functions that they perform in these patients. So having seen an association between the gut and response, we then wanted to look at the tumors and see if there is any association between the diversity and composition in the gut with what happens at the tumor site. So to do this, we first profiled the tumors in these patients and saw that certain type of cells, which are well known to be anti-cancer, were much higher in patients who respond versus those who did not respond. Now, this is not a surprise. This is obviously known, and this is what we expect, that responders would have a higher density of these anti-cancer CD8 cells. Our interest actually was in trying to correlate the bacteria in the gut with these anti-tumor cells. So if you can recall the Ruminococcaceae family, this family was implicated very strongly in responders. So on looking at genera within this family, we saw that there was a large cluster here that was very significantly associated with these anti-cancer cells. So there was definitely a link between these abundance of these bacteria in the gut and the type of cells that are present at the tumor and are attacking the tumor. So this plot basically contrasts a good bacteria, the Faecalibacterium, which was present in responders, and Bacteroidalis, which was present in non-responders, versus CD8 density. So you see here that in presence of the good bacteria, these cells are high, they are very positively correlated. Whereas in the presence of these bad bacteria, these cells are not so high. So therefore probably not resulting in a good response. So our take home from this was that high abundance of Ruminococcacea as well as Faecalibacterium in the gut was associated with anti-tumor T cells within the tumor microenvironment. So now all our findings were pretty compelling, but we also understood that this could be purely associational, as in we took patients who responded and we looked at their gut, we also looked at their tumor. So it probably might have been just because of that. So we then tried to understand the mechanisms behind these associations. And to do that, we performed experiments in mice. So these were germ-free mice. These mice lack any microbiome to begin with. They are completely devoid of microbiota. So they received transplants from either a patient here in blue who responded very well to therapy and had sustained responses over a long period of time, versus a patient in red here who did not do so well on therapy and had actually progressed on it. So upon receiving this microbiome, these mice now were carriers of the microbiome that was represented in the original donor. They were then injected with melanoma tumor, and these tumors were allowed to grow and then treated with drug to therefore monitor both the effect of the microbiome on tumor growth as well as on response to therapy. What we saw here was really striking in that the mice that got stool from responders grew much smaller tumors compared to mice that got stool from non-responders. They also responded significantly better to anti-PD-L1 therapy compared to the non-responder mice. And to confirm our prior findings, when we sequenced the stool that we collected from these mice, we again saw that the responder FMT group had a much higher abundance of Faecalibacterium, which was the bacteria that we were always implicating, to be higher compared to the mice that got non-responder stool. So in essence, the phenotype was being transferred from patients to humans, and one could now safely say that the microbiome was involved in not only influencing tumor growth, but also its response to therapy. So the obvious question is this just a melanoma effect, or is this also seen in other cancers? So as I had mentioned before, this is a highly active area of research, and parallel to us, a group at France did similar studies in lung and renal cancer patients, and they in fact strikingly found that patients that got antibiotics before beginning treatment with immunotherapy did not do as well on therapy as those that were antibiotic free. They also contrasted the microbiota in these patients, and they implicated the genus Akkermansia as well as the one that we found, Ruminococcacea, in patients who responded. And they saw that when this bacteria was given to mice, in addition to cancer and anti-cancer therapy, it greatly helped improve responses to anti-cancer therapy. Here are results from an investigator in Chicago who also looked at melanoma patients and who, like us, saw differential microbiomes in patients who responded versus those who didn't. And the bacteria that they implicated as well was associated with enhanced responses to anti-PD-L1 therapy. So all our work was recently published in the journal Science, and it leads us to the conclusion and several provocative questions that arise because of these studies. So there's certainly increasing evidence for the role of the microbiome in health and disease, and evidence that the gut microbiome may influence responses to cancer therapy. It also raises several important questions in the context of immunotherapy. Should we be profiling the microbiome of all patients going on to therapy? Should we be limiting or at least closely monitoring antibiotic use in their patients? Do we need to consider diet and probiotic intake in these patients? Do we also need to consider the role of the microbiome in preclinical models before drawing conclusions from them? So given that there is now this strong evidence to suggest that modulation of the microbiome can actually enhance responses to immune checkpoint blockade and other forms of immunotherapy, we now believe that this needs to be tested carefully in the context of clinical trials where immunotherapy is supplanted with some sort of microbiome modulator. So the answer to the question, can we modulate the gut microbiome to enhance responses to therapy, we believe is a resounding yes. These efforts have been possible through funding with the Moonshot. We've actually accomplished quite a lot in a short span of time because of their support. But importantly, there are also concerted efforts worldwide to better understand these phenomena and better understand these differences in bacteria that occur across cancer types and therapies, and try to build on these findings to improve therapeutic response. Importantly, we are also working with the Parker Institute of Cancer Immunotherapy, as well as Series Therapeutics, to implement a clinical trial, which we hope would open later this year, to test the hypothesis that modulation of the gut microbiome will enhance responses, using strategies such as fecal microbiota transplant, as well as other methods for microbiome modulation. So here are my acknowledgements. It did take a village to do this study. There were several different groups involved. First and foremost, I must thank my mentor, Dr. Vargo, who's in the audience here today, for spearheading these efforts, as well as all the wonderful people at MD Anderson who contributed to it. Last but not least, I must mention the patients and their families who generously contributed time and samples towards our cause. So thank you again for your attention, and I'm happy to take any questions if you have them.
Thank you, Dr. Gopalakrishnan. That is worlds of food for thought. Now, I'd like to remind those of you in this room that you have a small button there at your desk, which is a microphone. So if you will, is there a slide that shows that? Anyway, you know how to operate them. I think you've been here before, most of you. But you just press the button if you want to ask a question. And that will allow everyone, both here and at the remote locations, to hear your question. Those of you at the other Houston area locations will check in with each location to see if you have questions. And those of you who are watching remotely, or if you have a question once you leave today, please email your questions to mycancerconnection@mdanderson.org. Now, Dr. Gopalakrishnan will take your questions.
Thank you. Yes.
I was taking probiotics. Recently, I saw a lot of articles about not taking probiotic. So I guess I know that we're in the testing stage right now on probiotic. What would be your recommendation?
Yes, you're absolutely right. It's still in the testing stage. We do not have a conclusive result yet to say, but our recommendation would not be to do either. We need to test this in the context of a trial where we test the efficacy of a probiotic in the presence of immune checkpoint blockade and see if the probiotic is actually causing harm versus whether it's causing any benefit. So I would hold off on doing anything till those results become more confirmed. I guess so, yeah. Sure.
Deal with.
Right. Right. So, you bring up several valid points here. The environment certainly affects it. So, for example, if you compare it, so our findings were slightly different from the findings of our collaborators up in Chicago. So probably there were differences in the diet here versus the diet up there. Also, there might be geographical and lifestyle factors that may contribute to this effect. So yes, this is not a one-size-fit-all phenomena. There are definitely other factors that contribute to this effect, all of which need to be considered. Now, having said that, we are poised to answer these questions because from our cohort, we also collected this kind of information from our patients. We asked them what kind of food they eat, what their general, we collected their depression scores, their general lifestyle factors. So we will be able to answer these questions pretty soon. And then about your question on, oh, I'm sorry. So the question was, is there a link between the gut microbiome and conditions like Parkinson's, which are mediated through the release of dopamine, and as well as is there an association between the environmental factors and the results that we see? So I was just reiterating that yes, there is an association between the environmental factors and the type of response. And in addition, the gut-brain axis is being very extensively studied as well, both in humans and in mice. And in addition to Parkinson's, there have been associations seen even with autism. So yes, that's definitely a possibility.
The beginning of my career, I was in Mexico City, and I was with the one of the institutes
I see.
Of medicine. And they maintained that medical center that I was in, there were 250 pathogens in the atmosphere.
Oh, okay,
So
I see.
I mean, I got the immunity for all my life. I mean, this was 50 years ago, and I have a lot strong immunity.
Right. Thank you very much for sharing that, because now we are appreciating the importance of understanding what these pathogens do. So, yeah.
I think we have a question in Sugarland. Would you please ask your question now?
Okay. What can patients in treatment now do to improve their microbiome?
So, good question. Thank you. But I mean, we still don't have a definitive answer to that. We're still trying to find out the answers to that. We would, the bacteria that we implicated, it thrives in the presence of a fiber-rich diet. So, but again, none of this has been tested in the context of a trial. So we wouldn't recommend any type of microbiome modulation before actually testing it. What our results show is that the microbiome is closely involved. So the next step is to identify ways in which we can modulate it.
What is the thought on the effect of colon cleanses that some people are tending to do right now on the microbiome?
If I heard you right, you asked what the influence was on colon cancers. Am I right? Colon cleanses?
Cleanse.
Oh, I see. I'm not completely aware of that, though I do know that colon cancers, in fact, were one of the first type of cancers in which the microbiota was studied, and it was found that one particular bacteria called Fusobacterium nucleatum that was implicated in poor responses. So there have been a lot of studies trying to eliminate that and improve responses to cancer therapy, colon cancer therapy. I know I'm not answering your question, but.
Anyone else? Yes, sir.
Is there any association between having a good BMI, body mass index, and in order to increase the good gut bacteria, and what kind of foods patients like me can follow?
Right. Again, that's a valid question and a highly active area of research. There have been very close links between obesity and high BMI and microbiome. We did not have the power to test that in our cohort. We do have that information though, and we are poised to test it in larger cohorts. In terms of the types of diet that you can take, again, this needs to be tested and tried in a trial to be able to answer definitively. but the bacteria that we implicated were those that thrive in a fiber-rich diet. So beans, legumes, these would help in those bacteria to thrive. So that's the implication that we can draw based on our findings. But again, this has not been tested and needs to be tested with an actual means of microbiome modulation to make this assertion definitive.
And am I understanding correctly that it's better to have a better gut prior to immunotherapy, or does it matter? I just started last week, and I'm not sure whether I have a good gut or not. Yeah.
Yeah, no, all our samples were pre-treatment. So yes, definitely at baseline, all our samples were collected before a patient started therapy. So yes, we would definitely recommend having a good gut before therapy begins. But I mean, it doesn't hurt to have one throughout as well. So. Yes.
Congratulations.
Thank you. Thank you very much. So it was the early release was in November, but it came out in print in January. So it's January 5th, 2018. Yes. Yeah. Hi. Of course.
I apologize. My question is, what is the recommendation for those who don't have colons due to whether it be cancer or ulcerative colitis or any patients that do not have a large intestine?
Right. Again, that's a very valid question, and just for the benefit of the remote audiences, the question was, what about patients who do not have a colon? How do we characterize their microbiome? So we cannot characterize the gut microbiome in these patients. Though we don't, even for our collections, we were using the stool as a surrogate. So we would probably continue to use stool as a surrogate, though it may not be exactly representative of the microbiota. That being said, we could always characterize other sites such as the oral cavity or the skin, or like maybe direct swabs of the mucosa to see what bacteria there are. Yes.
If I want to know how is my gut, where do I get it tested? Who do I ask for? What, doctors would do something like that.
So there are a lot of private entities now that afford that service, where you can ship your sample to them and they would sequence your microbiome for you and tell you what kind of bacteria there are and what these bacteria do and what you may need to do to shift that composition towards a more favorable one. I mean, we do it in-house, but we're not yet we don't do it as a patient service yet, but we do it for research purposes. Yeah. I can look it up and pass that information along. So, yeah. Yes.
Kind of following up to this lady, so I'm going to be starting the microbiome through my oncologist. Do I understand that we will get the results whether we have good or bad bacteria or not?
So it depends on the protocol, whether that kind of how the protocol is set up, as to whether that kind of information will be released to you or whether it is just for research purposes. But I mean, these kind of studies have been outsourced to private companies who will be willing to release the results to you. So.
Again,
yes.
I will get the last comment. Because I have commented that before, the best data that you can get is between the two borders. I mean, the Mexican and United States border and the United States-Canada doing studies in relation to the public health. And this procedure to many of the activities because they have different physics.
Right. I mean, that's an excellent point, sir. And we couldn't agree more. We know that we strongly believe that there are geographical as well as race differences. We did look for that within our cohort. And so our cohort was not big enough to be able to test that, but others have shown that these kind of factors affect how a microbiome is shaped. So.
Thank you so much, Dr. Christian.
Thank you.
And let us thank him for his wonderful presentation today.
Thank you. Thank you.
Thank you. And thank all of you in the audiences everywhere for participating and helping us all further our own understanding of this crucial area. We hope you found the session informative and that you now have a better understanding of Anderson's efforts to help address survivorship needs. Please be sure to complete the questionnaire that is at your place to evaluate this particular presentation. And please come again to another picnic. Thank you for coming.